Human body is the most complicated system in the whole universe. You're made up of billions of cells. Current estimates put the figure somewhere between 10 billion and 100 billion cells. Inside nearly every cell is a nucleus, containing your own unique set of 46 chromosomes. Each of these chromosomes consists of a compact coil of an incredibly long molecule of deoxyribonucleic acid, DNA. DNA is a nucleic acid that contains the genetic instructions used in the development and functioning of all known living organisms and some viruses. The main role of DNA molecules is the long-term storage of information. The information in DNA is stored as a code made up of 4 chemical bases: adenine, guanine, cytosine, and thymine. DNA sequencing is the process of determining the exact order of bases adenine, thymine, cytosine, and guanine in a piece of DNA. As we can see, the shape of DDNTP, or deoxynucleotide triphosphate, contains the hydroxyl group in the third carbon. But the dideoxynucleotide triphosphate, or DDNTP's, molecules differ from DNTP by having a hydrogen atom attached to the third carbon rather than the hydroxyl group. The fifth carbon of an incoming DNTP is joined to the third carbon at the end of the chain. But when the DDNTP combines, the replication will be halted. In Sanger's reaction of sequencing, 4 things are needed: a strand to be sequenced, DNA primers, which are short pieces of DNA that are complementary to the strand which is to be sequenced, a mixture of a particular DDNTP with its normal DNTP, and the DNA polymerase, which is an enzyme that helps catalyze in the polymerization of deoxyribonucleotides into a DNA strand. As an example, the DATP will be taken. There are 3 steps in a sequencing reaction. In denaturation step, we will raise the temperature up to 94 degrees Celsius. When raising the temperature, and due to kinetic energy, hydrogen bonds between the strands break down, and they are separated, generating 2 single strands, as we can see now. Then, annealing step starts, where the temperature is cooled down to approximately 50 degrees Celsius. This is where only primer strands can anneal. Now, the primer is wriggling around until it finds its complementary strand and combine with it. Once there are few bases built in, the ionic bond is so strong between the template and the primer that it does not break anymore. The extension step starts, where the temperature is raised gradually to approximately 60 degrees Celsius. This is the ideal working temperature for the polymerase. The polymerase catalyzes the replication process, and this replication continues until the DDNTP molecule combine and halt the replication for the same reason we discussed earlier. And because the DDNTP will attach to every chain at different locations, we will have different lengths of chains, which can be separated according to length. Since the DTNTPs are fluorescently labeled, it is possible to detect the color of the last base of this fragment on an automated sequencer. At the end of the incubation period, the fragments are separated by length from longest to shortest, and this is done by the electrophoresis process. DNA molecules are set upon a polyacrylamide gel, where an electric field is applied on it, and induces the nucleic acids to migrate toward the anode due to the net negative charge of the sugar phosphate backbone of the nucleic acid chain. This separation of these fragments is accomplished by exploiting the mobilities with which different sized molecules are able to pass through the gel. Longer molecules migrate more slowly because they experience more resistance within the gel. Because the size of the molecule affects its mobility, smaller fragments end up nearer to the anode than longer ones in a given period. After some time, the voltage is removed and the fragmentation gradient is analyzed. Then, the sample is put to the sequencer. A simple DNA sequencer will have one or more lasers. The frequently labeled fragments that migrate through the gel are passing a laser beam at the bottom of the gel. The laser excites the fluorescent molecule, which sends out light of a distinct color. That light is collected and focused by lenses into a spectrograph. Based on the wavelength, the spectrograph separates the light across a charge-coupled device camera. Each base has its own color, so the sequencer can detect the order of the bases in the sequenced gene. Then, the bases are shown on the screen of the computer as a chromatograph, allowing them to know the DNA sequence.